everybody, my name is Patricia Soblecero Martin, and I'm going to talk about the pharmacological prospects in the treatment of Duchenne muscular dystrophy. I've chosen this topic because a couple of years ago, I received a Ortega Fellowship, it's a post-residency fellowship, and during these two last years, I've been working in the clinical laboratory service here in the hospital in Basurto, but also combining this work in the neuromuscular disorders lab at Biocruces Vizcaya Health and Research Institute. In my work there in the neuromuscular disorders lab, I've been performing a PhD project focusing in the evaluation of new therapies for Duchenne muscular dystrophy, and also in the development of gene editing techniques as a potential therapy, but also as a tool to generate better disease culture models. So I'm going to start making a review about the disease, Duchenne muscular dystrophy, which is a rare X-linked lethal neuromuscular disease caused by a variety of mutations in the DMD gene. This gene codifies for a protein called dystrophin, which is an essential protein to maintain the skeletal muscle structure. As you can see here in this picture, dystrophin binds the actin cytoskeleton of the skeletal muscles with this protein complex in the membrane. In Duchenne patients, during contractile activity and leads to absence of dystrophin, it causes sarcolemma fragility and also fiber damage. These fibers are eventually replaced by adipose and connective tissue, and also by clusters of inflammatory cells. Patients are typically diagnosed when they are in their early childhood, when they manifest their first signs of muscle weakness, like motor delay and difficulties to walking. With the disease progression, patients lost ambulation and the capacity and needs a wheelchair. Around puberty, they start with the disease progression. Around puberty, they start suffering cardiac and respiratory complications, and they finally die very prematurely. Today, there's no cure for Duchenne muscular dystrophy. The only treatment that have shown to delay disease progression are corticosteroids that are the standard of care, together with some multidisciplinary medical and surgical and rehabilitative strategies. But many therapeutical approaches are being investigated, and today I'm going to show you some of them. First, I want to talk about another disease related with this one, Becker muscular dystrophy, which is also caused by mutations in the DMD gene. But the phenotype of these patients are very mild, and they present a later onset and a slower progression, and also a nearly average lifespan. The different spectra of these two diseases can be explained by the open reading frame rule. According to this rule, mutations that are out of frame prevent the ribosome to reading the amino acid sequence correctly, and this results in the truncation of dystrophin protein. We have a total absence of dystrophin protein, and this happens in DMD patients. But sometimes patients have in-frame mutations, and with these mutations, retain the reading sequence normally, but the dystrophin synthesis is shorter. So we have shorted but functional dystrophin protein, and this is related with the milder disease. So as I said, there are many therapies in development, but as you can imagine, with this variety of mutations and having into account that the DMD gene is the largest human gene known, you can imagine the challenges to find strategies for these Duchenne patients. Some of them are focused in dystrophin restoration. I'm going to show you some related with read-through, exon skipping strategies, and also gene replacement and CRISPR-Cas technology. Another strategy is Eutrophine Modulation, and is the strategy that we have implemented in our laboratory. We have been working with this strategy with CRISPR-Cas gene edition, but also with small molecules. And the other thing will be find drugs to mitigate the secondary pathology of the disease. But today I'm not gonna talk about these ones. I'm only gonna focus in dystrophin and eutrophine. So the first one of eutrophine restoration would be the read-through therapies. Read-through strategies are applicable in DMD patients carrying nonsense mutations that are approximately between 10 and 15% of patients. Nonsense mutations are point mutations that lead to a premature stop codon in the mRNA, abolishing dystrophin expression. But these read-through compounds can recognize the premature stop codon and encourage the cell to ignore it and continue with the normal translation. So with this therapy, we restore the full-length dystrophin protein. Today, there's only one therapy conditionally approved by the European Medicines Agency. It was approved in 2014 and is Ataluren, Translarna, also called Translarna. The other strategy is the antisense oligonucleotide-mediated exon skipping therapies. This skipping approach is based in restore the rhythm frame in DMD patients that carry out-of-frame mutations. As in the example before, this will be a patient carrying an exon 52 deletion, and you can see here how the open reading frame is broken, and we don't have a protein dystrophin expression. But with antisense oligonucleotides that are small pieces of modified RNA, we can skip the machinery of the cell can skip the exon adjacent to the mutation and restore the reading frame. In this case, like in Becker muscular dystrophy, we will have a smaller but functional dystrophin protein. Today, there are 4 therapies conditionally approved by the Food and Drugs Administration, based in this approach, that are these 4: eteplirsen, golodirsen, bitolarsen, and casimirsen. But one of the problems is that there are very specific to the mutation of the patients, and they only are applicable for a small percentage of the patients, as you can see. So a promising therapy that has been in development, and it would be benefit for all DMD patients, regardless their mutations, would be the gene therapy that aim to restore dystrophin. And this strategy relies in the administration of exogenous microdystrophins or mini dystrophins, which are miniature versions of the dystrophin gene, lacking the unnecessary domains, and are coding into adeno-associated virus. The reason to create these miniature dystrophins is that dystrophin gene, as I say, is the biggest human gene known and is 500 times bigger than the vector. So that is the reason. And then this vector can be administered intravenous or intramuscular, and the vector, as you can see here, can goes to the nucleus and release the dystrophin gene, and this can be translated and dystrophin can be expressed. But we are talking about microdystrophins. It's not full dystrophin. Some phase 1 and phase 2 clinical trials are ongoing, and the preliminary results are very good because the patients are showing, as you can see here in these pictures, after treatment they show dystrophin expression in their muscle fibers, and also by Western blot these microdystrophins can be quantified. But although these good results in utrophin expression, is not yet clear whether or not this treatment will ameliorate the disease progression. And also, some patients have experienced severe adverse events related normally with an immune system activation. And the last strategy in dystrophin restoration I would like to comment is the one based in CRISPR-Cas9 gene edition. As you probably know, CRISPR-Cas9 is an adaptive immune system from bacteria against viruses that was discovered by Francis Mojica, but its potential application for to treat gene disorders was proposed by Emmanuel Sampentier and Jennifer Doudna. They received the chemistry Nobel Prize in 2020 for this reason. And this mechanism is compounded by two components, two essential components. One is the Cas9 is a nuclease that can perform a double-stranded break in the DNA, and the other is the single guide RNA sequence. This sequence can bind to our target gene and guide the nuclease to perform this break. After this break, the mechanisms of the cell can restore the sequence by non-homologous enjoining or by homology direct repair, in case we also include a donor DNA. So with this strategy, we can remove mutations and include the corrections in the genome. This strategy has been in preclinical studies, in cell culture studies, and also in animal models of the disease, have been investigated in hundreds of studies. I show you here a couple of studies. This one is performing an MDX mouse, taking skin fibroblasts, and after reprogram these cells to iPS, they perform a break here in this exon to knock out this exon and remove an out-of-frame mutation and recover the open reading frame of this protein. So after this correction, they induce differentiation of the cells, obtain myogenic progenitors, and they transplanted the cells again to the mice with very, very good results, recovering the dystrophic phenotype of the mice. This also have been performed in dog cell models, but using adeno-associated virus as a vector to inject the Cas9 and the guides, the two components in the cells. And as you can see here, the recovery after treatment of dystrophin expression is very, very, very important. But these studies are also preclinical because they have three risks. One is the presence of target events. Another is that we have to improve the delivery of these genome editing components into the cells, and muscle is a very complicated tissue. And also another risk is the activating of the immunoresponse against the Cas9 and this vector also. And then I'm going to start talking about the Eutrophine Modulation, because my thesis project and my work in the neuromuscular disease lab has been focused in eutrophine protein. Eutrophine is a dystrophin surrogate that could compensate the lack of dystrophin in patients because patients with Duchenne and Becker and also the animal models of the disease naturally overexpress eutrophine in their cells. And we think that could be a mechanism to compensate the lack of dystrophin. A lot of research groups are developing new therapies around this strategy. And the last year we published this article, is a review summarizing all these new therapies in development related with eutrophine overexpression. Some of them have been, for example, the eutrophine gene therapy. Eutrophine gene therapy development has followed similar pathways as eutrophine gene therapy. The groups have performed micro-eutrophins and developed and using this micro-eutrophin packaged into adeno-associated virus as well. And a lot of studies in MDX mouse and also in dog models have been published in the last years with very good results and a lower risk of immunoresponse adverse effects. But most of the studies focused in eutrophine modulation has been performed using high throughput screening of drugs libraries to identify small molecules that are able to induce eutrophine upregulation acting through the eutrophine promoter. You can see here the promoter and all the different pathways that are being investigated. And in blue, you can see some of these small molecules. The first orally bioavailable eutrophine regulator was sutromid, that after great preclinical research reached clinical assays, but unfortunately did not provide any significant benefit for the patient, and finally the program was abandoned. But here below eutrophine, you can see here another component that is being investigated now. Is a second generation compound related to sutromid that share the mechanism of action but has an improved physicochemical properties and also a more robust metabolic profile. But apart from targeting the eutrophine promoter, you can there's have been also investigated new therapies focuses in protein stabilization of the eutrophine protein to the membrane, and also in the mRNA stabilization. In this way, I would like to say that in our lab, in the neuromuscular disorder lab, we have generated a CRISPR-Cas9 gene edition mechanism to overexpress eutrophine by targeting this sequence, because this sequence is the target of a lot of microRNAs that inhibits eutrophine expression. So at DNA level, we have been able to remove this target of microRNAs and overexpress eutrophine in a cell culture line. Here I'm going to show you what have we done. What we did was to design two GFP plasmids, two plasmids carrying a fluorescence signal. And these plasmids contained both the Cas9 to perform the cut here and here, and the single guide RNA to guide the Cas to this place. As I say, this place is an inhibitory microRNA region, and by removing this region, we wanted to overexpress eutrophine in this cell culture model. We use DMD immortalized myoblast from a DMD patient. We transfect with these two plasmids carrying these two guys the cells. And then the GFP positive cells were single cell sorting in microplates. And after expansion of these cells, we analyze them to confirm or not the presence of this cut, if we have removed or not this region of interest. We perform first a genomic PCR, a normal genomic PCR, and the expected size amplicons were analyzed by Sanger sequencing, and we confirm in some of them that we have that we removed these target regions that binds microRNAs. Afterwards, we decide only to continue further analysis with only one clone. It was this clone 8, and we called it a DMD UTRN model. In this model, we evaluate eutrophine expression using 4 different techniques. Here is the Western blotting assay. This is the immunochemistry. And we have also used two novel therapies that have been implemented in our laboratory, MyoBlot and digital droplet PCR. Using all these techniques, we conclude that our model overexpresses eutrophine compared to the DMD patient cells, and we could conclude that it's a good proof of concept of a possible therapeutical option, but also that this model could serve as a positive control to perform in vitro screening of another eutrophine modulator compounds. So, using this model and two novel techniques, as I say, MyoBlot and ddPCR or digital droplet PCR, we perform this eutrophine quantification platform to help in the evaluation of therapies for Duchenne muscular dystrophy. On one hand, you can see here the MyoBlot. The MyoBlot allows us to quantify eutrophine expression, the eutrophine protein expression, directly in cell culture. In green, you see the eutrophine signal. And it's a quantitative immunofluorescence assay that shares the specificity of a Western blotting, but allow us to high throughput because we have a lot of number of wells to perform screenings. And on the other hand, we decided to implement the digital droplet PCR because it presents some advantages compared with the traditional method, the quantitative PCR. Like, for example, that it allows absolute quantification of dystrophin gene expression with an improved precision, sensitivity, and reproducibility. Thanks to this platform, we have collaborated with other laboratories and also other companies in the evaluation of eutrophine-based therapies. For example, with this one, Some Biotech, this company identified a repurposing compound chemically similar with ethutramide and contact us for evaluation with our platform. We perform a first screening of 60 compounds using MyoBlots and identified, as you can see here, identified 9 hits for further analysis. In a second collaboration with this company, we amplified the study using digital droplet PCR in four different compounds, and some of them, these two first ones, as you can see, have some concentrations showed eutrophine overexpression. So we were very happy with these results. But also in this time, in these 2 years, we have been also focusing the identification of new targets for eutrophine modulation. For example, we are very interested in this pathway focused in the promotion of the oxidative phenotype of the cell that directly acts through the eutrophine promoter. Studying this pathway, we identified an histone deacetylase inhibitor and proved it in our platform in vitro using DMD cell cultures by myoblood and also by ddPCR. And after obtaining very good result, we perform a very small in vivo assay in collaboration with the Neuromuscular Disorders Laboratory in Biodonostia using MDX mice. But unfortunately, I cannot show you these results yet because we are still analyzing, but we are very happy with them. And hopefully I will present them very soon in my thesis project. And this has been all our work during these 2 years, and I hope you like it. And I would like to finish. I aiming all the residents that finish in the hospital to apply for this kind of fellowships because they allow us to combine your clinical work with research. And if you are interested in research, these kind of fellowships are a great opportunity. Thank you very much for your attention.